thanks to Andrea and Manoli and the other organizers. It's fun to come back to the RECOMB conferences. The talks have been fantastic this year. As geneticists, we tend to think about and talk about and obsess about the genetic architecture of complex traits. But in fact, genes are the drivers, but phenotypes are the manifestation. And what we actually measure for the most part, and what we actually care about, and what evolution cares most about, are the phenotypes. And with that small change in perspective from genotype to phenotype, we get new insights and different perspectives on the kinds of questions that we ought to be thinking about. Typically, we worry about the number of genes, the function of the genes that underlie complex traits, the interactions between alleles, between genes and environment. Interactions are hard, but they're extremely important. And then we worry about how all of these genes work individually and collectively within the context of networks. But from a phenotypic perspective, we also worry about expressivity, how strong, severe the phenotype is depending on the combination of genes and the environment. Pleiotropy, the various manifestations of particular genetic variants. And then from a medical perspective, we call those comorbidities. And we also worry about the plasticity, how plastic, how fixed a given phenotype is, to how robust it is to variation in the underlying genes and to the environment. A lot of our thinking about complex traits is driven by Fisher and other pioneers of genetics who focused on traits that were more complex than what Mendel focused on, things like height. This is the medical school class, I think at Yale back in the 30s or 40s. They're lined up on the yard lines of a football field according to their height, the women in white coats, the men in blue coats, and you can see a nice normal distribution. You see substructure in the population where men and women differ in their heights. There's a newer photo of this, and if I could ever find it, it would be cool, because they redid the photo a couple years ago, and everyone has shifted, not because of the underlying genetics has changed that much, but because of environmental effects. The average height of everybody now is taller, but the same underlying statistical distribution of populations. So Fisher and others, we study populations because, as Willie Sutton was asked once, why do you rob banks? That's where he answered, that's where the money is. We study populations because that's where the statistical power is. But we lose a lot of interesting genetics and biology by averaging effects across the population. We'll come back to that point in a bit. But Fisher and others also made some simplifying assumptions at the time that the phenotype is determined by individual genetic effects, environmental effects, but also gene-gene and gene-environment interactions. And to make their mathematical analytical life simple, they assume that gene interactions are basically zero, that over a population, the average effect isn't too important, and the gene-environment interactions aren't too important. And in a way, we've forgotten that those are assumptions that need to be tested, that need to be explored. As hard as they are, they're still out there to worry about. This is another way to think about it. There's actually another factor involved. If you think about the growing worldwide epidemic of obesity, diabetes, and other metabolic conditions, these phenotypes result from genetic variants in your DNA, from lifestyle and what you eat and how much you exercise, but also epigenetic effects that translate both environmental inputs and genetic inputs into the ultimate phenotype. So there's actually another important class of variation in addition to genetic variants and phenotypes that finally determine your phenotype. So I'm going to talk about three short stories, three topics today. The first deals with fractal genetics and gene discovery. This fractal genetics is a concept that Eric Lander and I discovered while working on the chromosome substitution strains. About epistasis, problem that Eric and I had actually written. You can do many things with chromosome substitution strains, but one thing you can't do is study epistasis. And part of the fun in science and research is that you have these fixed positions, fixed beliefs. It turns out the chromosome substitutions strains are exactly the way to study epistasis, and the evidence for them is remarkable. So it's fun letting nature tell us where we're wrong. And finally, I'll end with some examples and points about transgenerational effects where epigenetic changes can be inherited from one generation to the next. So All of this will be in the context of metabolic disease, but if you happen to be interested in cancer or bone biology or neurologic conditions, every time I say metabolic disease or anything related to that, just translate that into your own particular interest. So this is David. You all know David. David went on sabbatical to the US for a year. This is David on his way home. David's DNA is the same, but his environment was different, and he made some lifestyle choices that presumably affect his biology and perhaps the next generation. And the challenge in metabolic research is understanding how to prevent weight gain, or once people are obese and have metabolic conditions, how to reverse the traits. And with the substitution strains, we can get at both the genetics, physiology, systems biology of weight gain, but we can also get at weight loss too, the really neglected and important problem in humans. We talk about weight gain, but there's a lot of people who are already obese, already susceptible, already show metabolic diseases, and we'd like to understand the reversibility of those conditions. It's really hard in humans when you combine genetics, environment, and epigenetics and heterogeneous populations. So mouse models are especially important. And years ago, Richard Surwitt picked 2 strains at random, AJ and Black 6. They weren't selected to be anything. It's like taking 2 people at random in the room, cloning you, and being able to do the human equivalent. You can't do that in humans, so we do this in mice. On a low-fat diet, Black 6 is the light blue line, and AJ is the light red line. So they're both lean. They're eating this high-fat diet equivalent to 100 days of Big Macs and supersized colas. So imagine if we did the experiment with you over 100 days, that's all you could eat, as much as you want, whenever you want, but that was your diet. On average, your physician is right, don't do it. But if we did the experiment, we would find that on average, many people would show diet-induced metabolic conditions. But invariably, there will be people in the room who remain lean and healthy. Doesn't seem fair, but it's the case. But here's AJ on the high-fat diet, and there's Black 6 on the high-fat diet. So only Black 6, only on the high-fat diet, shows diet-induced obesity. AJ, regardless of the diet, is lean. Now, what's interesting is the 2 things your physician would tell you is eat less. In fact, and so you would expect AJ to be the light eater. In fact, AJ eats 40% more per day than Black 6. And your physician would say, well, okay, if you're going to insist on eating the diet, get some exercise. AJ is the mouse equivalent of a couch potato. It eats, pees, poops, and sleeps. And Black 6 spends his time running around the cage. So there are genetic examples in mice of these various conditions. So we wanted to get at traits like this. And years ago, Eric and I anticipated that as the Genome Project went along, we would have ultimately genetic variants that span the genome so we could finally get at the genetics of complex traits. We imagined that most genetic variants would have phenotypically small effects, that we would find some of these variants, but not all of the variants because of statistical power. The variants that had big effects we could see above the noise, but a lot of interesting and important variants would be buried in the background noise of genetic studies. And we imagined the same would happen in mice, that we would get some of the variants, but not all of the variants. We guessed that someday we would discover this thing called missing heritability. And so we thought that we could create a resource that was different from all the other resources that had been made. And humans were obligated to study genetic variation in a heterogeneous population. And if we do crosses in mice, we have the same thing. What we thought we would do is substitute individual chromosomes one at a time from one strain to the other. And what we would create a chromosome, oh, I missed this. Let me go back a second. I jumped ahead. At the same time on the high-fat diet, Black 6 mice also shows insulin resistance, hypertension, heart disease, non-alcoholic steatohepatitis, and diet-induced liver cancer. And so everyone studies the Black 6 mice to get at the genetics of these conditions, diet-induced conditions, to get at the genes and mechanisms, and ultimately figure out treatments and prevention. AJ is resistant to all of these conditions, even though they eat more and run less. And so Eric Topol and I think about this as the genetics of disease, and this is the genetics of health. Nature has developed a solution to these problems, and AJ may have the answer. So I think both of those are important. So here's the chromosome substitution strains. We took Black 6 and AJ, and through repeated back crosses between the strains, we replaced each of the autosomes in Black 6 with a corresponding autosome in AJ. It took 7 years. We genotyped 17,000 mice. But we finished. Now we have 22 strains, new inbred strains, that differ from Black 6 by a single chromosome. So then by testing this genetically uniform population for diet-induced conditions or any other phenotype of interest, and comparing each of those 22 strains to black 6, or multiple testing penalty is only 22, we can ask, is there a genetic variant, at least one variant on the substituted chromosome that affects the trait of interest? If these two are the same, then we either say there's no genes or the genes are weaker than our statistical threshold. If there is a difference, then we know that there's at least one gene on the chromosome, and we can begin to do gene discovery and functional studies with that strain because it's genetically defined. We can do as many studies with genetically identical animals as we want. So what we're doing is not changing the signal, but reducing the noise. Each chromosome is tested on a uniform, identical, and reproducible background. So we put the chromosome substitution strains on a high-fat diet, the same protocol, eat as much as they want for 100 days. And here's black 6 again, here's AJ, and you can see that at least 18 chromosome substitution strains differ significantly from black 6, which means each of them has at least one gene, which means there's at least 18 genes that control response to diet-induced obesity. There are 4 strains that are indistinguishable from black 6. At the time, most of these genetic variants weren't discovered. There was a recent paper in the Collaborative Cross looking at this kind of problem, and they found 1 QTL in the genome survey. At the same time, the next issue of Genome Research, we published a paper in a single 20 megabase interval. We found 14 QTLs and identified 2 of the genes. So the genetic complexity of these traits in a heterogeneous population is really hard to dissect. So here's a quick summary of some important points before we get into the other points that I want to emphasize. Many CSS's substitution strains have QTLs. We've looked at more than 90 traits and located more than 700 QTLs for an average of 8 QTLs, or CSS's or QTLs per trait. These CSS's and the corresponding QTLs have unexpectedly large effects. From Fisher and Wright and all the genetics that has been done before, we imagined in complex traits that individual genetic variants would have small effects. And the phenotype then is the sum of all those small effects. And in crosses in mice, Jonathan Flint and others wrote a review a few years ago, the average QTL effect is about 6% of the total difference between the parental strains, the total variation. In the CSS crosses, the average across those more than 90 traits and 700 QTLs is 76% of the parental difference. In other words, a single chromosome substitution almost completely recapitulates the full phenotypic difference between the parental traits. The other interesting feature is that 92% of the substitutions switch the phenotype from the black 6 state not away from AJ, but towards AJ, as though the 2 strains are bistable states. They can be like AJ and they can be like black 6, and we substitute a chromosome. It largely moves it almost all the way towards towards AJ. But I want to dig into one chromosome a bit because it tells us an important point. It emphasizes an important point. We picked a single chromosome at random, a single chromosome substitution strain, the CSS6, chromosome 6, not because we knew anything special, not because it was special in any other way, except it was the first strain ready to go for these studies. So here's black 6, here's AJ, here's chromosome 6, here's the weight, the BMI, the body mass index, and the fat pad weight. And you can see that AJ differs substantially and significantly from black 6 for all of these traits. And here's chromosome 6. It's almost as big effect as AJ. So this strain is resistant to diet-induced obesity. It's one chromosome. The other 17 chromosomes are other stories. So the way we can dissect this then is to take that single chromosome and through recombination and in just a few crosses, we can fragment that chromosome into smaller pieces. We call these congenic strains. So here's the black 6 bit of chromosome 6. The rest of the genome is black 6. But now we just have this little segment of chromosome 6 near the centromere derived from AJ. A longer segment, still longer, 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 longer. We had a panel of 17 or 18 of these. I'm only showing the quick summary of this. So we put the congenic strains on the high-fat diet for the same 100 days and asked where's the gene on chromosome 6 that confers resistance in such a strong way to diet-induced obesity. So here's the strains again. Here's their names, just lab code. Final body weight, difference in body weight from black 6 or from the previous strain, the p-value, and the phenotype. Now this study was done 3 times in 2 different mouse facilities with genetically identical mice. So we're really confident about these results. It's a value of these strains, the attribute of these strains, that we can reproduce these studies so well. So the first strain is equivalent to black 6. So we say there's no gene in this little interval. If we make that interval longer, these mice weigh 7.2 grams less than 92A. There's the p-value. So there must be a gene in this little interval that confers resistance to diet-induced obesity. If we make the segment a little longer, this mouse is now 8.8 grams fatter than that mouse. There's the p-value, and it's obese. And as we go down the chromosome, as we increase the length of that segment, the phenotype toggles between obese, lean, obese, lean, obese, lean. This is not unusual to diet-induced obesity. Everyone else who's been studying these strains for whatever trait they're interested in largely finds the same results. So many QTLs, large effects, and contrasting effects. So then we took this strain, because it had the strongest phenotype, and we broke that into pieces, and we call those subcongenic strains. And then we took some of those and broke those apart into sub-subcongenic strains. So here's this strain, and what we thought was one QTL here, we called it OBRQ2, is in fact 1, 2, 3, 4 QTLs. So it's like those little Russian dolls. You open up one and there's more, and you open up another, there's more. Here, one QTL is in fact multiple QTLs. And if we then take this interval and dissect that into sub-subcongenic strains, we find 1, 2, 3, 4, 5, 6 QTLs. So in this one 20 megabase interval that we started with, we found 14 QTLs that have significant effects on diet-induced obesity. What's interesting, too, is that the parental strain differed in glucose, insulin, and obesity. And when we get down here, we can find this one affects obesity, but not glucose and insulin levels. This is obesity and glucose, but not insulin. This is glucose, but not insulin or body weight. So this phenotype is not inherited, controlled in an integrated way. It can be teased apart into each of its components. So not only are there multiple genes, there are also embedded phenotypic differences. So that's the story for one little bit of one little chromosome. Across all the QTL discoveries we've done, where we've tried to dissect the complex trait in this kind of way, we summarize the data in this. And this is what Eric and I call fractal genetics. Fractals are where you have similar patterns of variation at whatever level of resolution. I don't have to explain fractals to you guys. So here's the AJ strain, the substitution strains, congenic, subcongenic, sub-subcongenics. This is the interval. The whole genome, a single chromosome, is on average is 120 megabases. The congenic strains on average were 28, the subcongenics 15 megabases, and the subcongenics 1 megabase. Now this might sound like a lot of work, but in fact it was one project of many that a postdoc and a student were doing over a year and a half. So it demonstrates the feasibility of dissecting these traits in a robust way, in a feasible way, with a small group. This is the number of genes in the genome, the average number of genes in the substitution strain, congenic, subcongenic, sub-subcongenic. So on average, we're able to, with simple crosses, to dissect an average interval that's composed of 4 genes. When you get to 4 genes, you can imagine all kinds of genetic engineering and manipulations to figure out what those genes are. It's a feasible number. There's a couple interval that have only a single gene. So we're pretty confident. And both strains have been completely sequenced. So we know all the sequence differences. But look at the effect size. 100% of the parent difference. The average was 75, 76% for the substitution strains. The average effect for congenics, subcongenics, sub-subcongenics. So what we've done is a 3,000-fold change in resolution from the whole genome to this sub-subcongenic interval. The number of genes was reduced 5,000-fold, but the phenotypic effect was only reduced 2.5-fold. It didn't dissect the way we imagined. As I said, we expected a lot of genes with small effects. What we're finding is a lot of genes whose effects seem unexpectedly large. Okay. The other little point is we did a cross with chromosome 6, substitution strains in black 6. So now our genome is a single chromosome because the rest of the genome is black 6. And we could do a conventional linkage analysis to ask where the genes are. And you can see the centromere, telomere, there's a log score curve, there's lots of peaks, there's lots of genes. The one interval on this chromosome where you could claim from this analysis there are no genes is right here. Except that's where OBRQ and OBR3 are, the 2 genes with the biggest effects on the chromosome. Typically, in a linkage analysis, you're hoping, in a GWAS2, that the variants are sufficiently separate from each other that each give you a signal. If the density of genetic variants is sufficiently large and they act in contrasting directions, here they're so close together we don't get enough recombinants to see the individual effects. When we went back into the data and asked for the few recombinants that separated those, they had the phenotypes that we saw in congenic strains. But together, there's not enough of these exceptions in the population to give a sufficiently big signal. Okay, given the complexity, can we find the genes? I'm not going to go into the evidence, but we found two of them. This gene, a solute receptor, affects body weight and glucose homeostasis in the endoplasmic reticulum. And this gene is expressed in axon, at the nodes in axons in the periphery and controls the localization of channels that control potassium going in and out of cells. So despite the complexity, we can find the genes, and in both cases we found genes, identified genes, provided functional evidence for the genes that have properties we wouldn't have expected. You would never expect that solute receptors that are involved in protein glycosylation would be important, or that localization of a channel on an axon would be important. Okay, that's the first part, fractal genetics. And as I said, we see these properties across all the traits that we and others have looked at. The next part is epistasis, and if you paid attention to the last little bit of evidence, you'll realize the importance of the next point. Not only are there a lot of genes, but too many of the substitution strains and too many of the QTLs have too big effects. In fact, 8 of these substitution strains are indistinguishable from AJ. In other words, that single chromosome substitution is sufficient to switch the black 6 state to the AJ trait. But there's 8 different ways to do it. So Eric and I, and typically it's implying non-additive effects, implying that epistasis is common in these strains. Now typically we look at epistasis by pairs of gene-gene interactions, a pairwise analysis. And we don't have statistical power to do that very well. And not only do we have trouble with pairwise, higher order interactions are almost impossible to dissect. And that on the basis of that argument, that's why Eric and I had written, we can't study epistasis in these strains. But given this evidence for pervasive epistasis, pervasive non-additive effects, we thought maybe we could do another kind of analysis to quantify the patterns of interactions. So what we did was to take the phenotypic effects in their natural units and add them up. And the argument is that if they're simple and additive and independent, we should be able to add up the effects and get somewhere from 0, no effect, to be able to explain 100% of the variation, right? And if it's Fisher, if it's conventional, we should get somewhere near 100, and we've fully enumerated the genetics, we should get close to 100%. So this is the sum of the traits, the variants, the CSS effects, for 42 traits. We're just adding up the values, and then we propagate the error so we can estimate the variance. And as soon as one passes the error around the mean effect, the cumulative effect, passes 100%, we say that it must be non-additive. So this is, I'm going to get into what I mean by this in a second. So if the sum is less than 100%, we say it's additive. If the sum is more than 100%, it must be non-additive in whatever order it is, whether it's pairwise, 3-way, 4-way, it's non-additive. Of the 41 traits, 40 were non-additive. The cumulative effect is 803%. In some cases, we could explain almost 1,400%. So while we have missing heritability in GWAS, we have excess heritability in the chromosome substitution strains because we can detect gene interactions. Chromosome 6 is cholesterol levels are interesting. 9 different chromosomes affect cholesterol level on a regular diet. The average effect of those 9 is 100% of the phenotype of the parental difference. In other words, each of those chromosomes is sufficient to switch the cholesterol level from black 6 to AJ, to lower it. We don't have any drugs that have effects that big. That's interesting. The other interesting point is AJ has all 9 of those variants, and it's no lower than any one substitution strain that has a single AJ chromosome. Another example of non-additive effects. I said I'd take this apart. This was the combined. We put together those CSSs that had significant differences, and just for the fun of it, we looked at the rest of the CSSs that had non-significant effects, thinking maybe those effects all act in the same direction. When we sum these, we're keeping the sign of the effect. If it increases, there's a plus. If one decreases, it's a minus. So these are additive and going in the, non-additive and going in the same direction. So even among the non-significant CSSs, many of them show cumulative effects that are non-additive. This is really good evidence about the pervasiveness of non-additive effects. But now we can go back to the populations and ask, what's the genetics and molecular biology systems properties that lead to this non-additivity and this context-dependent effects? So how do we reconcile this? Fisher is right about, and others are right about populations, the average effects, the average genetic variance. Jonathan Flint in his summary was right, and across the average phenotypic effect is 5 or 6%. But the substitution strains give us a different picture. I think the issue is, in a population, we're averaging effects across a heterogeneous background. In the substitution strains, it's like getting enough statistical power for the first time to make a confident statement about the genetics and phenotypes of a single individual, a single genotype. And because they don't overlap, these are independent statements from each other. Okay, the model that we get that comes from this, and I haven't shown you all the evidence for this, but it's a summary of it, that epistasis is pervasive, that organisms are composed of non-random combinations of genetic variants that we largely can't detect, but in mouse there's some other evidence that supports this. And these specific combinations confer sufficient viability in breeding so that the strains persist. Epistasis buffers systems from perturbations like diet or any other, or even genetic variants, we'll get into this in a second, and allows us to survive. We're not just a bag of genes, we're a network of interacting genes that confer good health and viability, but also buffer against perturbations. And the disease then can be thought of as disrupting these networks of gene interactions. Okay, the last part, this is the most, the other parts are a little bit unconventional. This part is really unconventional. So Mendel, a lot of our concepts in genetics have been driven from the work that Mendel did. People wondered for a long time about inheritance. Mendel was the first and most important breakthrough. By choosing simple traits that were inherited in a simple way, he was able to ascertain the rules that guide the relation between genotype and phenotype. And he's looking within an individual that some inherited factor that we now know as DNA and genetic variants accounts for simple phenotypes like the color of the petal or the height of the pea plant. That Mendelian principle of associating genotype and phenotype is what we do in GWAS. It's what we do in any association or linkage study. We take a population of obese or lean or cancer susceptible, cancer lean, and try to associate genetic variants in the affected versus non-affected population to get a clue to the genetic variation that accounts for phenotypic differences. So it's the foundation of most of modern genetics. And in that context, the work, gene discovery work that we've been doing, that metabolic disease and other cancers, is what drove us too. But in the course of this work, we discovered something that didn't make any sense until we thought about it in a different way. So we all know, and there's increasing evidence, that environmental factors like famines during the Second World War in Netherlands, in Sweden, in Greece, led to phenotypic effects of the mother not having sufficient nutrition while she was pregnant that adversely affected the fetus. And when the fetus grew up as an adult, it showed increased susceptibility to metabolic disease, neurologic conditions, as though there was a metabolic memory. Even though good diet and environment had been restored, the individual remembered the conditions under which they were when they were gestating, and that left an epigenetic memory that guided their phenotype later. And we know that in some cases that phenotypic effect can then be transmitted to next generation. The epigenetic changes can be heritable. And that changes the rules a whole lot. Your phenotype isn't just you, your genes you inherit and the environment you inherit, but it may be the cumulative effect of what's happened in generations past. So I want to tell you about transgenerational genetic effects, not just environment, where genetic variants in previous generations affect your phenotype, even though you don't inherit that gene. In other words, we could do this survey, like a GWAS survey, and have variation in affected individuals and find some of the genes because they inherited those genes, but some of the genetic effects may be inheritable epigenetic changes induced in previous generations by risk genes that are then inherited to the next generation. So the trait is genetic, it is heritable, it is familial, it has all the feelings of the statistical properties of inherited traits, except the individuals who are affected haven't inherited genes. They inherited the epigenetic changes. So I'll give you one example. Here's OBRQ2. As Dave and Soha broke the congenic strain into a subcongenic strain, they took this interval, and there was a 3 megabase interval here that had some genes of interest. I'm not showing you the other two. Those two intervals also had QTLs of interest, but they wanted to look at this one. Here's the difference in body weight between BLAC6 and the 161 congenic strain, or subcongenic strain. Here's insulin levels, here's glucose levels. So Dave and Soha wanted to test for parental effects, for which there was no evidence in the way this was inherited. There were no genes in the interval that are known to affect epigenetic features like methylation or histone changes. They just had this feeling, a suspicion. This is fun of working with students. Sometimes often they have clever ideas So what they did was to cross BLAC6 with the 161 strain. So the mice then, the resulting hybrids, are heterozygous for this relatively randomly selected 3 megabase segment derived from the AJ genome. The rest of the genome is BLAC6. When they intercross these, the way Mendel would have, you reconstitute BLAC6, you reconstitute the congenic, and they threw away the hybrids. They weren't really interested in those. And if Mendel is right, if this is inherited in a conventional way, the reconstituted 161 should be lean, the reconstituted BLAC6 should be obese, and it assumes that this heterozygosity for this 3 megabase segment in the parental generation doesn't have any unusual effects. So remember, BLAC6 should be obese, 161 should be lean. It's important to emphasize that all of these mice were maintained and bred on a regular diet. All we did from that breeding colony when we wanted to test was to move some of the animals to a high-fat diet and test whether they inherited the susceptibility. Here's BLAC6. Again, each dot is a mouse, 3 different studies in 2 facilities. There's the average. Here's the reconstituted 161 strain. Here's the reconstituted, here's the parental 161. There's the reconstituted strain. So the 161 strain behaves correctly. Here's the reconstituted BLAC6. Instead of being obese, it's lean. So then Dave and Soha took these mice and intercrossed them. They're genetically, they're pure BLAC6, right? For the next generation, they're still lean. They went one more generation before we left Cleveland for Seattle, and they're still lean. So their phenotype is not determined, can't be determined by their own genetics, it's determined by previous genetics of previous generations. We have other examples. Oh, one thing. This is inherited through the paternal line, not the maternal line. So it's not simply a maternal effect on the physiologic effect on the fetus. Other examples, the father's Y chromosome can affect the daughter's phenotype, even though the daughter doesn't inherit the father's Y. Otherwise, she'd be a son, not a daughter. And testicular cancer, we have lots of examples. These effects are strong, they're persistent. They can be through the maternal lineage or the paternal lineage. It means that your phenotype is determined both not only by your own genetics that you inherited in a Mendelian way, the environment to which you're exposed, but also by the environment and genetics of previous generations. It means that some of the variation that you inherit, this variation, these epigenetic changes, aren't embedded in DNA sequence differences, but are result from other molecules that we inherit. And obviously identifying those molecules is really, really important because it means that our phenotypic information is not only in DNA, but also in some other molecules that remain to be identified. So I'm running out of time, and I'm going to skip a bunch of these. Okay. These are the folks that did the work in the lab and the collaborators. I just moved to Seattle not long ago. I'm looking for postdocs, so if any of you are interested, we have really cool projects. I haven't emphasized the computational aspects, but they're behind everything that we did. And I'll finish there and answer questions. Yep. You mean the variation?
I think the robustness is different environments. So when you have a lot of genes making you obese, you are robust in any case.
Well, but
only one or two, you might be less robust to variation in the environment.
so if you were, maybe a way to think about that, I'll have to think more about your question, but one way to think about it quickly is if you look at the effect size, it doesn't scale with the number of genes. The effect size remains large. It's independent of the number of genes in the interval. Perhaps that's what you're getting at.
That's
And maybe
the mean.
That's the mean effect.
But the robustness would be if it's stable to variation.
Yeah. That's an issue that we think about a lot. If you look at some of the strains, and I didn't show this, some of the substitution strains have huge variation, some have small variation, and it's reproducible. But to get at the genetic control of that robustness, that genetically defined physiologic space, is really hard, because you have a lot of evidence on which to base an estimate of the mean. But to do the statistical analysis of variation is harder. So I believe it's there, but I don't have any, I have anecdotal evidence, but I don't have proof. But it's something we think about a lot. And I haven't talked about some of the other evidence for this. But there is evidence for buffering and robustness. None of the strains were bigger than AJ. Few, if any, were lower than different from black 6, as though the parent strains defined the range of phenotypic variation, another measure of robustness. So it's there. And maybe the substitution strains are a way to tease it out, but we haven't quite figured out how to do that yet.
Hi, thank you. Your results are very striking. I was curious, from what you've learned, would you change anything about the way we do GWAS and select populations for GWAS?
It's hard to change GWAS because it's conditioned on the populations in which you study. If you embedded in a lot of what I said was context dependent, like that OBRQ2 and OBRQ3. OBRQ2 has a really, really strong effect, almost as big as the whole AJ genome, but its effect can be completely hidden by another variant. So in that little population that had OBRQ2, we'd be fine. We could see it. But you change that with a little bit longer segment or something else in the genome. So as we gain statistical power, so to go back to GWAS, as we gain statistical power to detect more genetic variants, we're averaging across even bigger populations with maybe even more genetic heterogeneity. And so we lose the ability to detect idiosyncratic variants that act in a context-dependent way. We increasingly ask for the common variants across a larger population. So in a sense, you're stuck. And maybe we have to transition from doing GWAS the way we're obligated because of the population characteristics to a dialogue with animal models and go back and forth. Manoli?
I mean, just as a follow-up question, the ability to now map more and more rare variants within these should potentially reveal some of these, fractal aspects. But I don't know if these have been there in the initial studies. I mean, perhaps we just don't have the sample sizes in order to detect these modifier effects.
It could be that what we're seeing in GWAS, what we're probably seeing, are genetic variants that have relatively strong effects on average that's largely independent of genetic background. What we can't have difficulty seeing are genetic variants whose effects are context dependent. So I think about you could imagine that in most populations, under most circumstances, the genetic variant is there, but its effect is small. But depending on genetic background and environment, that effect could be big. So I call these context-dependent signal and context-dependent noise. But you're stuck. It could be a rare variant, not because it's a variant that's rare in the population, but its phenotypic effect that we can measure is rare. The variant could be common, but the phenotype could be rare.
For the third part, when you're looking at the mapping of these sort of transgenerational loci, how much of the variance do you explain by these loci, and does that give you any hint about the number of loci involved? Because if you explain a large number with just a small region, that suggests that the regulation, in a way, is very simple, whereas a lot of us in this room might expect it to be more complex.
So we don't know the genetic. We know, for example, in testicular cancer, that if we use genetic variants in particular genes, and therefore it gives us clues to this RNA biology rather than histone modifications and DNA methylation in the gametes, the effects are pretty strong. As strong as the susceptibility alleles that we've been characterizing. But David Altshuler, when I presented this at Broad, said, Joe, this is really interesting, but there's small decorations on the core phenotypes that are inherited in conventional ways. And that was partly the reason why we looked at the Y chromosome effects, because the daughters don't inherit the Y. So that's one of the reasons. But the other is that it let us compare chromosome substitutions to the daughters. And it turns out we looked at 120 traits, and the daughters, even though they're genetically identical to black 6, and their only difference from black 6 was the father's Y, the same fraction of proportion of the 120 traits differed phenotypically, and the phenotypic effects were as strong as substituting a whole chromosome. So they don't appear to be rare, and they don't appear to be weak. Mapping and understanding the molecular biology of it's important,
Yeah.
but it seems to be as common and as strong as conventional inheritance,
Fascinating.
which is pretty scary
Thank you very
and pretty
much.
cool.
So let me ask one final question. Since you like to be provocateur, let me be provocateur. Why does it have to be the gene? So why can't it be the structure of the chromatin that actually, because that seesaw effect that you saw was very striking.
Yeah. I absolutely think that that's at play here. I talk about it as genes just because it's a simple way to think about more complex issues. But you're absolutely right. It doesn't have to be the genes. And my guess is
Genetic profiling to show whether, for instance,
we want to do that. An example of the evidence for that 161 congenic that showed the parental effect on obesity through the paternal germline. Remember, that was embedded within the OBRQ2 interval. We then Dave and Soha then went back and tested the parental, the congenic strain. It doesn't show transgenerational effects. So that interval is context dependent. Its ability to transmit a heritable epigenetic change depends on the chromosome segment around it.
thank you, Joe.